In a previous video, we have seen that structural lipids can assemble into membranes to form the physical barrier between a cell and its environment, or between compartments within a cell. The membrane is also organized in functional domains with specific lipid composition and a specific composition of bound proteins. In this video, we'll try to better understand how lipids and protein diffuse in a membrane. In this animation, we see an example of these functional domains called a lipid raft, which is thicker than the rest of the membrane because it's enriched in phospholipids and contain a cluster of proteins that have a very specific function. As you can see, both lipids and protein can diffuse in the plane of the lipid bilayer. How can we measure the lateral diffusions of lipids and protein in the membrane? We can use an experimental approach called Frap, or Fluorescence Recovery After Photobleaching. This is a relatively simple experiment. The first step is to label the surface of the membrane with fluorescent molecules. So for example, you can label all the lipids at the surface of the membrane. Then you expose a small region of the membrane to an intense laser light that will bleach the fluorescence. So now you will see a dark spot surrounded by a sea of fluorescent molecules. And you can measure the time it will take for this dark spot to become fluorescent again. This recovery of fluorescence is due to the lateral diffusion of labeled lipid from the surrounding region into the dark spot. And you can create a graph showing how long it takes for the fluorescence to be recovered. From that graph, you can determine a diffusion coefficient of the molecules, in this case the lipid, that was labeled. This diffusion coefficient is in fact determined based on the radius of the bleached spot and the half-time of fluorescence recovery. Once you have this diffusion coefficient, you can, using a very simple equation, determine the average distance traveled by a labeled molecule per unit of time. For example, a lipid will travel 2 micrometers in 1 second. To give you a sense of what it means that a lipid will move from one side of a bacteria to the opposite side in 1 second, or from one side of an epithelial cells to the opposite side in few minutes.
Do membrane proteins diffuse as fast as lipids?
Not exactly. The rate of diffusion of proteins in a membrane is quite variable. Some proteins, let's say for example a photoreceptor in a rod cell from the retina, diffuses as fast as lipids. In contrast, in red blood cell, the chloride bicarbonate exchanger diffuses very slowly. Why is that? This exchanger is a transmembrane protein and has a region, the intracellular region of this protein, interact with a dense network of protein that is located just beneath the membrane of the red blood cell. This network is called the membrane skeleton, and the tethering of the exchanger to this network prevent its or limit its lateral mobility. Now, what are the functions of membrane proteins? We can make two observations. The first one is that the total concentration of protein in a membrane varies from cell to cell or from organelle to organelle. For example, in the inner membrane of a mitochondria, you have about 70% of protein. That high concentration of protein is in direct correlation with the function of the inner membrane of mitochondria. The inner membrane of the mitochondria has to transport electron during oxidative phosphorylation from one protein complex to another. And this membrane also contains the protein that is necessary for the synthesis of ATP. In contrast, Schwann cells have very little protein in their membrane. And that is also connected with their function. Schwann cells act as insulator. And this insulation property is mainly due to lipids and not to proteins. The second observation is that you have also a difference in the protein composition from one cell to another cell or from one organelle to another organelle. For example, red blood cells have a large variety of protein in their cell membrane. In contrast, the rod cells from the retina have also a lot of protein, but it's basically one type of protein, the photoreceptor. And so in a way, the membrane performing different functions will have different repertoires of protein. In other words, the protein composition of a membrane reflects its function. So how do protein associate with membranes? We can take the example of the membrane of adipocytes. You remember that when adipocytes are activated, a catecholamine binds to a receptor. This receptor is embedded in the membrane of the adipocyte. It is called an integral membrane protein. The portion of the receptor that passes through the membrane is hydrophobic and interact with the core, the hydrophobic core of the lipid membrane. Now on the other side, on the cytoplasmic side, this receptor interact with a trimeric G protein. This G protein is not embedded in the lipid bilayer. It is called a peripheral protein. And you have peripheral proteins both inside and outside of the membrane. In the case of the trimeric G protein, its beta subunit interacts with the other subunit and is attached to the membrane via non-covalent bonds. In contrast, the other subunits of the G protein, the alpha and gamma subunits, form a covalent bond with lipids that are inserted into the lipid bilayer. These are also called peripheral membrane proteins. So we have seen that protein, membrane protein, can be involved in the transduction of a signal from the outside of the cell to the inside. But membrane proteins will also be responsible for the permeability of membranes. Membranes are semi-permeable. The lipid bilayer is impermeable to ions and polar molecules. For example, potassium ions and sodium ion cannot move, they are quite small, but cannot move through the lipid bilayer. Ions and small polar molecules will traverse the membrane via membrane proteins. And these membrane proteins will be either pumps or channels. The control of the membrane permeability has also important functional consequences. For example, in the inner membrane of the mitochondria, the impermeability to proton is extremely important to create a gradient of proton needed for the synthesis of ATP. The opening or closing of protein channels that allows the transport of ions across the membrane are extremely important for the transmission of action potential along neura. Water is an interesting exception. It is polar, but can move through the membrane very easily. There are two reasons why membranes are permeable to water. There is a very high concentration of water in the environment of a cell. There are also channels that allow the diffusion, the transport of water across the membrane. These channels are called aquaporins. And water molecule will move across the channel in a central pore by interacting with the protein surface that line this internal pore. Aquaporins can be very specific. They can allow the passage of water molecule, but not the passage, for example, of small ions like potassium or sodium. There are aquaporins that are a little bit less specific. We've talked about one of these aquaporins. These non-specific aquaporins can allow the passage across the membrane of small polar molecules like glycerol in adipocytes. In conclusion, in this series of videos about lipids, we have described two critical roles for lipids: the energy storage and production, and the formation of membranes. Lipids play also an important third role. They play a role in signaling both within a cell and across cells. And we'll see examples of lipid-based signaling during the rest of this course.